So yeah, I'm going to talk to you mostly about fish schooling, but I'll sneak in a few other species that relate very strongly, even if at first it may not seem so. So I'm really fascinated by how interactions bring minds together. So there's a lots of people that study the individual brain, but there's far less research trying to understand these invisible connections among individuals that give rise to collective behaviors, such as in schooling fish. But more broadly, what I'm going to talk about today is spatiotemporal computation, what brains have evolved to do to make sense of a very complex world. And I'm largely going to focus on fish schooling, just because of this audience. I think it might be of more interest than some of the other systems that we study, but you'll get a sense of the diversity of species that we look at later on. And because I'm talking about fish, I get to talk about stuff I hardly ever talk about in my talks. I just don't have time to talk about it all. So today I'm going to talk about biomechanics first. So it's been long held belief that fish may benefit from hydrodynamic interactions when schooling. However, there's a big problem with this assumption. And that's the main problem is that there's no evidence. Right? And why is there no evidence? Well, if we think about the theory that was developed by Danny Weiss back in 1973, he made the prediction that if this were the case, then fish should be in this lattice formation to recover energy from those behind. And then relatively, a few years afterwards, in 79, another paper was published in Nature that showed for the multiple species that they looked at, you just don't see this rigid formation. Fish are moving with respect to each other in a more fluid manner. And so it turns out to be very challenging. Hydrodynamic interactions are hard to measure for fish where they're expected to be important. The Reynolds number makes it very difficult. We humans, despite our amazing abilities, can't solve turbulence yet. And so Liang Li joined my lab as a PhD student, a visiting PhD student, then continued to be a postdoc and has just started his own lab in Konstanz, so he's now a PI. And he said to me, well, what we can do is develop robots that swim like fish. And even though we can't ask how much energy a fish is using, we can ask how much energy the robot's using. And the problem with fish is if you put two fish in a tank, they're less stressed than with one fish. And so there's going to be independent metabolic reasons why you get differences for different group sizes. But robots don't get stressed, at least not contemporary robots, maybe in the future. And so we can use robotic fish. Firstly, we can quantify that they produce the same vortices that fish use, but we can directly measure how much energy do you use. And we did these experiments. So if you did this on a supercomputer, this would take years probably. But here, just using actual real systems and measuring these properties, we could find that in actual fact, what mattered wasn't really how far the fish were apart, at least within the tank we used. What mattered is really how far they are between the front and the back. And the red area is bad. That's where they're losing energy. They're paying a cost, and the blue area is where they're actually gaining energy from their environment. And so what you should note here is that the phase difference. Now, what that means is if the phase difference is 0, that means the fish are in sync. They're swimming like this. If the phase difference is maximal, they're swimming exactly out of sync. But of course, you can have something in between. And what you note is as the front-back distance changes, if the fish just adjust their phase difference continuously as a function of their position with respect to a leader, it doesn't matter that they move around. They can always adjust to optimize energetically. So the physical experiment revealed a hidden but very simple rule. That's cool, but of course, you want to understand, well, why is that happening? And so the hypothesis is that individuals can benefit from socially generated flows irrespective of exact position. So this algorithm's kind of hidden in plain sight. We didn't know what to look for before prior to these robotic experiments. And then we wanted to understand why is this the case. And I won't go into details here, but we used direct measurements of the fluid, putting little particles in the water and using lasers to track the particles. We used supercomputers to conduct simulations of the fluid flow, and we developed what's called an analytic model that allows us to prove certain properties. So let's ignore lots of details and just think about the large-scale flow properties in the fluid. And this just represents this continuum between computational fluid dynamics and a simplified version that's analytically tractable. And what this was able to show is that the theory could also predict, so the theory are these dotted lines, the theory would also predict correctly what the phase difference is that optimizes your transport with respect to the leader. And it turns out to be relatively simple. So the vortices shed by a fish's tail induce a flow in the fluid behind it. And if you look at D, for example, so the vortex has already passed, but there's more fluid moving in that direction. And if the fish moves its tail in the direction of that fluid, it can save energy. And similarly, if the vortex more directly, or the interaction between vortices as shown in E, moves in that direction, the fish can recover energy. But fish may not always want to recover energy. What if you want to move faster? Well, what you can do is you can go out of phase, and then you kind of use these vortices like a ladder to push your way through the environment more quickly. But on average, we expect that fish would utilize this to save energy. And we call this vortex phase matching. So now we have a theory that is irrespective of spatial position. As long as you're close enough, we can then test this with real fish. Are real fish following this behavioral algorithm? And so we have fish in a flow tank in Konstanz, and we're tracking the fish, and we can record properties such as the left-right distance, the front-back distance, and so on. So for every moment in time, we can ask, what does the theory predict the follower should do? And then we can ask, what does the real fish actually do? And so here you can see the theory in black and the experimental data in dark gray. And so you can see the fish tends to track what the theory predicts. That is, the fish are using the predicted behavior that we found from the robotic experiments and the modeling. Now, when we do the modeling, of course, it's an idealized version of reality. So the data look incredibly clean here as you change the phase difference, and it wraps around because this is a circular variable. But the important point is if the fish weren't doing this, this would just look gray or some sort of intermediate color. You wouldn't see these stripes. Those stripes are the fish adjusting its phase to the individual. And if we look at the data, we see clear evidence of these stripes. And of course, we did statistics to show that this is a strong effect. So that's really cool. So it's sort of hidden in plain sight was a very simple rule that fish can and do use to save energy. But the next question is, how do they do that? Now, if you look at the literature on fish hydrodynamics, almost every paper, if not every paper, assumes that they use a system called the lateral line. It's a series of pressure sensing hairs. There are two different types of what are called neuromasts that sense the pressure and the flow of water. We've known this for a long time, that fish can sense flows in this way. And so people naturally have come to the assumption that this must be what they're using to sense flow, such as socially generated flow. But one of the great things about doing experiments is you can often get surprises, and I'll show you that in a moment. And of course, fish have another system that might be used for these high temporal dynamics, and that's the visual system. Okay, so how do we manipulate those to ask how they contribute? Well, no vision is easy. We can turn the lights off and see what they do in the dark. It's reversible. No lateral line is also, we want to minimize the harm to the animal. And it turns out you can knock out these sensory cells using antibiotics, and you can use microscopy to prove that you've knocked them out, and they will regrow so the fish are ultimately unharmed. And then you can have fish that have no vision and no lateral line. And so if the lateral line was important, we should expect to see it here. Now, without vision, they can still do it. So this is consistent with the lateral line being the sensory modality that's required. Without the lateral line, however, they can still do it. So maybe there's redundancy. Maybe you can use either vision or the lateral line. And that's why it's critical to knock both of them out, and they still do it, right? And so we now have evidence from neural recordings that they're almost certainly using proprioception for socially generated flows, and that frees up the lateral line system for paying attention to flows in the environment that matter. For example, turbulence and eddies that might be indicative of where food is, and so on, or the structure of the environment you can sense through flow. So proprioception, what is that? Some of you may not know, because it's actually less studied than many other sensing mechanisms. But I can feel, if I put my hand out of the car I'm driving, I can feel the flow because it deflects my body, and there are sensors in my joints that detect that deflection. Right? So the fish, its body, by being deflected, there are sensors along the spine that detect the deflection. And we think this is an active process, not entirely passive, because our neural data suggests that the neurons fire before the passive mechanism can set in. But anyway, the point to note is that they're feeling the flow that's socially generated, not using the lateral line. And we think this frees up the lateral line to then detect other important features. And we're now asking questions like, how do they remove socially generated flows when they're trying to detect environmentally generated flows, and so on. There's also practical applications to this knowledge. You can see here, if the fish swim out of phase with each other when they're laterally beside each other, they can produce more propulsion. But actually, if you think about fish schooling, they're not necessarily in it together for the benefit of each other. They can be genetically unrelated and therefore selfish. And it turns out that the hydrodynamics of schooling is a game theoretic problem. Yes, if they adopt the same phase difference in the middle, they do pretty well, right? You could see that in the previous video. However, if I am offset a little bit backwards from you, I can adjust my phase difference to cause a cost to the one in front to benefit me. Right? And so it's a game theoretic problem. And I've not seen any work thinking of fish schooling in this way. I mean, it's very counterintuitive, right? That the fish in front can be influenced by the one behind. But even if you're directly behind, the hydrodynamics of this one impact the one ahead. So all sorts of fascinating questions for the future. So fish could actually be moving with respect to each other to exploit vortices generated by each other. Or it may be a mutualism, that they've evolved not to exploit each other, but to be in that sweet spot where they all do okay, even if they could do better by cheating. So this is ongoing work. And we're also doing things like fancy tomographic particle image velocitometry that allows us, our theory before was developed in 2 dimensions, and it was good enough because the fish were swimming roughly in 2 dimensions. But of course, if we're dealing with more complex schools, we have to think about 3-dimensional flow. So things get much more complex, but we're working on that problem. We've developed software that allows us to remotely reconstruct the full 4-dimensional body posture, so 3D space and time being the 4th dimension. And we can also track the eyes of the individuals, so we can see who's looking at whom within the group. Now, if we want to study the hydrodynamics, what we can do is we can put these little particles in the water, these neutrally buoyant particles, and use laser sheets. The problem with that is even if you use infrared lasers like we do, the fish do not like it, right? And also the ethics, and there's lots of problems. Plus, you've got one time to look at a particular dynamical interaction because, of course, you can't reverse time to look at it again at more detail, for example. So what we do is we record the posture of the fish as they're swimming, and then we replay that exact time, with the same time scale, the same size fish, but in robots, in a robotic platform. And with robots, we can zap them from all sides with lasers. We can replay time again and again and again, look at different resolutions, so we can then better understand how the fish are interacting with each other. So we can replay the position and the kinematics, the tail beats of the individuals in our robo-twin platform. So it's a physical robotic twin of the real world that allows us to get very accurate information. And again, we can ask, how much energy do you use, and so forth. And so we have this ability to 3D print these robots, and we can measure very precisely the power cost, the thrust, and analyze the flows at multiple scales. In addition, we can track the eyes of the individuals remotely, the real individuals, of course, and reconstruct their visual fields. And we're also mapping the retina to see, are they looking in the gaps for predators? Are they looking at each other? How do they couple the two eyes when they're swimming together? And this is, again, ongoing work. And vision is very important. So I talked about the lateral line. Now I'm going to forget about it for the following reasons. These are golden shiner fish. We also work with modulation, which you also have here. And then later on, I'm going to talk about zebrafish, but juvenile zebrafish. These fish here, the golden shiners, have a very primitive lateral line system, right? So they are not using the lateral line for schooling. They probably use odor, but they largely use vision. And so what we can do here is we can reconstruct the visual field from the left visual field in red, the right visual field in blue for all of the individuals, but here I'm just showing one. And you can see this complex time-varying scene. And what we're interested in is, how does the fish brain, or I mean, this is a general question for all animals, how do you take in this complex sensory information and reduce it down to low-level movement decisions? And as was mentioned in the very kind introduction, we've done a lot of work over the years looking at things like collective sensing, studying, for example, how groups, this is a real group, by the way, this is a real fish school, and we've worked out which visual features they pay attention to so we can reconstruct this network of communication. And we can address all sorts of different interesting questions about collective sensing and collective decision-making. But because this is all published, I'm not going to talk about it today. You can chat to me afterwards. I want to talk about the stuff we're doing right now. And it really comes back to this idea. I mean, it was really by studying these collective minds, this collective behavior of animal groups, that I started having ideas that I realized were missing from neuroscience for what's going on in the collective within the brain of the individuals. And when I wrote this paper in 2007, I was very cautious to draw an analogy between collective computation within the brain of the individual and the type of collective computation I was looking at in animal collectives. The level of selection is completely different. The units, the entities are very, very different. But what I'll argue today is I was wrong. In actual fact, there are fundamental principles in common. So I want to take you on that journey now. To start, just giving you a quick overview of where I began and what we consider now classical models of collective behavior. How this then made me start thinking about the brain as a collective, of course, on the move through space and what we learned from that, and to some really recent work, published 2 days ago, in fact, where we're revising completely our understanding of fish schooling, insect swarming, and I'll show you that these early models that I developed are completely wrong, which is a wonderful thing to discover in science, and it makes new predictions about the brain. So let's start off with the classical models of collective behavior. So when I started working on this problem, I was really interested in what's going on inside the head of the individual that makes it behave the way it does. And because we had no experimental data at the time, I used computer simulations that speculated individuals interact locally with simple rules, as was mentioned in the opening. And what I speculated was, if individuals got too close to each other, there's this zone of repulsion where they would avoid collisions with each other. And because we saw alignment among the individuals moving in the same direction, we assumed, turns out completely incorrectly, that the animals were explicitly aligning with their neighbors, that they knew what direction the neighbors were going and they were copying those directions. I'll show you later that's completely incorrect for every system we've studied. And that there's a tendency to be attracted to each other, which allows groups to stay together. That definitely exists. But this was a starting point. And at the time, we produced patterns and properties that were reminiscent of what we see in reality. And so this led to a lot of research showing how local simple rules can lead to large-scale collective dynamics, which is, I'm very proud of and is very important, even though I'll argue today that it's fundamentally wrong. And so what I want to show you today is that when we consider a fish like this shark or any animal, we can't think of the brain as following these simple fixed behavioral rules. It doesn't make any sense. The brain has evolved to make decisions. Therefore, inherently, it is a dynamical system, right? Just like the fish school itself, by the interactions, leads to nonlinear dynamics, we also have nonlinear dynamics within the brain of the individual. And these old models just had simple rules. If the environment's like this, you do that. And what we argue is that we need to move away from that paradigm and explicitly consider the sensory-motor interface. I mean, this makes complete sense, right? You need to put the brain in or the cognition into collective cognition. But amazingly, 99.9% of the papers in collective behavior still use the rule-based models. So I want to talk to you about consensus decision-making, but within the brain. And then I'll come back to how this has changed my perspective on fish schooling at the end. And before I go further, I want to thank the many amazing people I've had the pleasure to work with. So Vivek was a PhD student with me who did, I'm going to show you fly experiments because we understand more about the fly brain, but don't worry, I'll get back to fish. Vivek did the fly experiments and hated them so much, he now works on large mammals, and he just got a faculty position at the University of Washington in Seattle, and he's going to do an amazing job there. Liang Li, I mentioned to you before, in this context, he did the virtual reality experiments with fish. Dan Gorbanos was a black hole physicist, and we got him involved in thinking instead of particles interacting, let's think of animals interacting. So he did the analytic models, so there's provable models. Bianca was an undergraduate who did locust experiments, and I've been collaborating with Nia Gov at the Weizmann Institute, who's actually a chemical physicist that works on cells mostly, but I've managed to get him excited by collective behavior at higher scales, and Mate Naj, who was a postdoc with me and has now set up his own lab in Budapest. And I want to show you this schematic, because I think this is one of the most remarkable discoveries in neuroscience in the last decade. So almost exactly 10 years ago, and I'll show you the papers in a moment, Vivek Jarayaman at Janelia discovered that there's an architecture within the brain of a fly that is literally a ring of neurons that encodes directional information. And I got super excited about this because I was always thinking about, in our sort of classic schooling models, of vectorial information, these directions of travel. And then in 2023, Ruben Portugues in Munich, now he's moved to Cornell University, discovered a convergent evolution of the same architecture in the fish. Now, so when we did this work, we didn't know the fish had this architecture, but we speculated it did based on the data I show you. And Vivek's work is really remarkable. So this is from one of the first papers. So the fly, this is a bump of electrical activity shown in yellow. And it's really telling you what direction what that direction is. And so let me show you a video from this early work. The fish paper, 10 years later, the data's cleaner. I'll show you that afterwards if you don't quite get the fly data. But before we go there, let's look at the ultrastructure, the neural connections. I mean, natural selection has found that structure. I mean, it's beautiful. And what I want to argue today, that all animals have to move through space and make spatial decisions. Therefore, we should expect this type of beautiful architecture to solve this complex challenge. And so here you can see the bump of electrical activity with respect to a virtual environment with a bar. And as you can see, when the fly turns, the neural activity turns. So it's like a compass in the brain that's telling the fly which direction it's moving with respect to some sort of external cue. Turns out flies like to use polarized light. They like to use the position of the sun. They like to use distant landmarks. So do fish. They use similar properties to know whether they're heading, for example, in one direction or the other. And so this is this beautiful paper by Ruben Portugues. Now, importantly, in the zebrafish brain, it's not literally a ring. The cells are in two bunches, but they're connected to each other, so they act exactly like a ring. And so let's have a look at his beautiful data from 2023. And so here you can see these two regions in the brain, and as the animal changes its heading, the encoding of that heading is very clear. So here you can see that bump of electrical activity changing as the animal moves through space, which maps very closely to its heading. So animals have a compass in their head. They're representing information on this ring as bumps of electrical activity. And so we've been doing a lot of work about, because I'm not going to talk about the compass. There's tons of work now on the compass in the last 10 years. So animals have a compass. They lock onto some visual feature. Doesn't mean I lock onto the same feature as you. I might lock onto the window at the back of the room. You might lock onto the screen, but it means you know where I am with respect to other objects in the room, rather than just with respect to yourself. We also know, and we know much less about this, and that's what I'm going to talk about, is how do animals actually establish a goal? Okay, so they have a second ring attractor that is used to establish where should I go? And then they use the compass to help them navigate. And we know that, if they have a goal that suddenly appears this way, a food patch appears this way, or another fish appears over there, and you want to move towards it, and my heading direction is currently this way, we know how the fly, at least, will turn towards its goal. So I won't go through that today. Let's just assume animals can turn towards a goal. And what I'm really interested in is how on earth do they establish a goal in the first place? How does the brain have this goal in mind? And also, if, for example, there are multiple fish around or multiple food sources, I don't want to get stuck in indecision. I want to decide between them. And how does that happen? And so I'm not going to go through mathematics at any point today, because I think everything can be explained through intuition. But I do want to note that the mathematics is very rigorous. We can prove all of these results. Okay, so it's actually a very simple model. So how does the ring attractor work? So again, it's collective dynamics. So we have this ring of neurons, and it turns out that you're connected with excitatory connections to the neurons close to you. So if you get activated, you excite them. If they get activated, they excite you. But simultaneously, you've got these long connections that inhibit other neurons that have a different opinion, so to speak. So they're competing with each other. They're sort of extinguishing opinions that are distant to their own and reinforcing their own opinion by positively communicating with neurons that are close to each other on the ring. And this is called a neural ring attractor network. So local interactions, again, in a sense, you've also got long-range negative connections, but simple rules that give rise to what the brain does. And so you can think of this local excitation and long-range inhibition, and the brain is coming to a consensus, because it's really, yes, you could probably get the brain sometimes to be not in consensus, and the animal doesn't know where to go, but this is very dangerous. So it's adaptive to actually decide, even if you have no information in the environment, you need to decide somewhere to go. Or you can stay stationary, of course. Right? And so one of the first things we've done, and it's the most recent work, it's in review at the moment, is ask, well, I'm going to mostly talk about decision making, but also animals, when they don't know what to do, or there's no sensory information, they have to do adaptive things like search. And if you look at the literature on search, there's things like what are called correlated random walks. There are things called levy flights that are thought to be optimal for certain decisions. Beautiful work by Rudiger Weiner in Zurich shows that ants, for example, if they can't find the nest, they will do a spiral search. Other animals do a zigzag search. So there's not an infinite number of patterns we see in nature. Right? Where do these patterns come from? No one has asked that question. People quantify the patterns. They can measure the statistical properties and say that's a levy flight, that's a correlated random walk, that's a spiral. But there's no work that's how does an animal actually generate these patterns? And so I won't go into details about this paper, except to say that hopefully it will come out in the not too distant future, where we can show that even without sensory input coming in, these neurons exciting each other and inhibiting each other, especially for anyone that knows about reaction diffusion and Turing patterns, local excitation and long-range inhibition is how you generate stripes or spots on an animal's coat. or an animal's skin, like the zebrafish, similar mathematics underlies the patterns that form on the ring in the absence of input that allow animals to adopt different patterns of motion. And this can be what's called systematic search, where there's a sort of systematic pattern to it, like looping search or zigzag search. And then there's a phase transition, a sudden transition to random search patterns like levee flights or correlated random walks. But surprisingly, I mean, one of the many things that's surprising about this is Rudiger Wener's spirals turn out to be a random search in terms of the neuroscience. Yet, if you look at the animal, it looks incredibly systematic. So we're very excited that this model can reproduce these types of dynamics. We want the simplest model that can explain everything we see in nature, right? And so this is looking at animal behavior without sensory input. What if there is sensory input? And that's what I'll focus on today. And so I want to re-emphasize the generality of this question. All animals have to make decisions on the move at some point in their lives. And I think they're going to be evolutionarily conserved computationally. And so we can show with the ring attractor, if you have two targets, so here, two stationary targets, what the model predicts is that the animal will average between these two options up to a critical angular difference, and then the brain exhibits what's called a bifurcation, a sudden change in neural dynamics. And it's a little bit unfortunate that it's called a bifurcation, because what I mean is, in dynamical systems theory, a bifurcation. It's also literally a bifurcation in space-time, in the trajectory, but that's sort of a coincidence, so to speak. What we really mean is that there's a phase transition in the brain. You know, if you cool down water, it's liquid. Cool it down a bit more, it's liquid. Cool it down a bit more, it's liquid. Cool it down a bit more, it's liquid. Cool it down a tiny bit more, it crosses 0 degrees, it's solid. That's what's called a phase transition. And collective systems exhibit these sudden transitions in collective state. What we're arguing here is that we're not cooling the brain down or heating it up, but as the animal, if it's far away from the targets, the angle is small. Therefore, the targets are inducing electrical activity on the ring attractor. And the ring attractor, they're sufficiently, the two bumps of electrical activity, I'll show you a simulation in a moment, the two bumps are close enough together that they can merge. That attraction is due to this excitement. They mutually excite each other. So they're close enough for the excitement to dominate. Excitement dominates, excitement dominates. But you get to a critical point where suddenly they'll fling each other apart. Right? So the brain is undergoing this phase transition. And I'll show you a simulation. So imagine you've got perception coming in. We look at vision, but this could work with acoustics, it could work with olfaction. And so as the animal moves through space, so if we start again when the animal's further away from the target before the bifurcation, then you see there's just a single bump in the decision-making layer. There's two bumps in the perceptual layer. They represent the targets, the stimulation into the brain as to where those targets are. And once they get separated enough, that single bump in the middle becomes unstable and the brain will suddenly switch. And if there's two equal strength targets, it's 50-50 which one they will choose, right? So this is a prediction of the model based on fundamental neurobiological principles. And so if we look at the analytic model, so this is, we can prove this result. I'm just going to explain exactly the same thing again, but just in a slightly different visualization. We're again always thinking about what's called the egocentric angle between the options. That's the angle between the options from the animal's perspective. If I'm far away from two options, that's going to be small. As I get closer and closer, say the two edges, the two corners of the stage here, if I'm over here, the angle's relatively small. As I get closer and I stand in between them, it's 180 degrees. So I smoothly go through this transition and we predict that the animal at the black line will transition into this collective state. I don't have time to go through this gray regime for now, but it's called a hysteresis regime, but let's just, I'll keep it simplified. So the argument is that we should see this sudden transition in the trajectory of the animal if the brain is doing what we think it's doing. So we want to test this theory in a wide range of animals. And later on, I'll show you that we could not find a single publication. Imagine how many publications there are on decision-making. Tens of thousands, possibly hundreds of thousands. We could not find a single publication where anyone had recorded how an animal moves as it makes a decision. People have looked at saccadic movement in the eye and reaching movements, but not the movement of the body. And so we developed immersive volumetric virtual reality, and I'll explain for several reasons why this is so important to control this. So we want to have highly controlled environments that the animals believe to be the real world, but we can confine them in the lab. And it uses a very simple illusion called the anamorphic illusion. And I can tell you right now that this tape does not have a volume above the table. You see, it never did, right? But your brain thought it did. And even if it goes back roughly into the right position, even I've told you this is an illusion, and you know it's an illusion, it now looks 3D again, right? So similarly, this shoe is not actually 3D. And so if you track an animal in 3D as it moves through 3D space, and you then use a computer 100 times a second or 200 times a second to reconstruct this illusion, the animal can be fully interacting in an immersive environment. And we do this with fruit flies, and we do this with locusts, and we do this with ants. This is unpublished, but later on I'll show you fish. I'll get back to fish, don't worry. Because it's all the same. That's the whole point I want to make. So we have this theory that we should see these bifurcations as animals move through space. We have the fact that we can't find a single experiment, so we had to do it ourselves. And here you can see the data from the flies. Later on, you'll see, this is published in 2021. Later, you'll see data from 2025 from ants, and it's way better. But this is how science works. And again, we see this bifurcation. So they're moving towards static targets. So you're looking down. They're moving through 4 meters or 1.5 meters of virtual space, and they're moving freely in this space. Now, the ants are not moving freely. They're pinned in place. But they're just so remarkable. So the theory predicts, for example, if you have 2 options that initially start at 30 degrees from your starting point on the left, all the way up to 180 degrees on the right, we can make predictions about the paths that animals should take, if our theory is correct. And here are the unfiltered data from ants as they make these decisions. So strong evidence across these different systems that the brain exhibits this bifurcation, a sudden transition in neural dynamics. And this is real data from the ants, from their perspective of this bifurcation. I can't believe these data. It's unbelievable to me how clean this is. And this actually, the student that's doing this, August, did his undergrad here in Zurich at ETH. Okay, so I'm excited about this because I want to understand how animals move through space. But there's a reason to be even more excited about this. Because what I haven't told you about is that any system that undergoes this sudden transition, a bifurcation or a phase transition, whether it's an ecosystem, whether it's the traffic in the streets, whether it's water turning into ice, close to the critical point, close to this transition, crazy stuff happens. Crazy stuff that we don't put in, that comes from the physics of collective systems, collective dynamics. And the most important of these, and this is exactly why we're so concerned about our planet, or for example, there's people here that maybe work in lake ecosystems. Lakes can suddenly transition from one state to another. And close to these transitional points, there can be wild fluctuations in system dynamics. There can be critical slowing down and properties like this. But what all this is indicative is that any collective system undergoing this type of transition becomes ultra sensitive. That's terrible for ecosystems because it means a tiny little bit of human influence can cause it to trip over into a different state. And then the hysteresis regime that I mentioned before means it's almost impossible to go back. This is good for decision making because once you make a decision, you don't want to go back to indecision, but terrible for ecosystems. The mathematics is exactly the same. And I can't take credit for this. This is this amazing Nobel Prize that, because we knew that collective systems seemed to behave in similar ways. Like a magnet, as you heat it up, suddenly losing its magnetism. Or traffic, suddenly there's congestion and no congestion. But Kenneth Wilson showed that the mathematics of this is universal, and these are universal properties and provable properties. So we can be pretty confident that in the context of neural dynamics, if the brain is behaving like this, then there's ultra-sensitivity, and the neurons are going to be super sensitive, just like the ecosystem is. The neural groups super sensitive to any differences between the options at the critical point. Amazing. That means that the little tiny brain becomes like a supercomputer at that critical point as an emergent property. And it doesn't matter the angle the animals come in at, we predict they'll reach this critical point and suddenly sort of see the difference and then make a decision, right? It's an amazing, really counterintuitive property. So it means that differences between options will be amplified at the critical point, and it's an emergent property. We're putting it in. It's so hard to explain. Like, there's so many weird things that happen at critical points. One side of the system effectively talks infinitely to other parts of the system, and time breaks down, and all sorts of cool stuff happens. And so we think this is an important principle, because we know if there's a difference between options and it's hard to make a decision, you can take more time. But for animals, in many cases, it could be costly to take more time. And this geometric bifurcation allows you to cheat that. The brain will emerge, as an emergent property, make a better decision. So if we have a preferred option, so say I'm over here, and I'm between these two options, and the one to my left is my preferred, and the one to the right is my unpreferred, and we randomize it in our experiments. Biologists often put an animal in between two options. Fish on one side, fish on the other side, put the animal in the middle, for example. But we predict there's a massive difference if you allow the animal to have this, what's called an embodied process. As the animal moves, it changes the geometry, which changes the neural consensus, which changes the movement, which changes the geometry. So it's called embodied cognition. And indeed, the locusts really struggle, but if we allow the brain to go through the bifurcation as predicted by the theory, it amplifies the difference. And for ants, exactly the same. And so what about, many biologists just do experiments with two options. But believe it or not, animals have to make decisions with many more than two options. And existing models of cognition do not scale. That's almost exclusively why psychologists do two options, because their models don't scale to 3 or 4. This model can scale to any number of options, as I'll show you shortly, but experimentally, I'll show you data from 3. And so I was astonished when we did this, because I thought the right option will cancel the left option and the animal will just go down the middle. This is the whole purpose of doing simulations and modeling, because it can show you counterintuitive things. I was wrong. The brain will actually spontaneously bifurcate and exclude the worst option, then the next worst option, and the next worst option, being ultra-sensitive at each bifurcation point. And so in order to see the bifurcation structure, if one of these options was different, the animal would just be seen to move towards it, and you wouldn't necessarily, I couldn't tell you which model is right or wrong. But if I make the models symmetric like this, I can see the bifurcation structure. Of course, the bifurcation structure is there precisely to discriminate. But in order to see it, the world needs to be ultra-symmetrical, which is why VR is so important. And indeed, for the flies and for the locusts, we found evidence that the brain is bifurcating in this way. And even recently, we found evidence that maybe even humans exhibit the same type of bifurcations. And as I mentioned before, this is an elegant fractal-like algorithm that means that you can make decisions for any number of options. And if, for example, you get to this option and it turns out to be a patch with many different options, then this process can just repeat itself at any scale. Okay, let's get back to fish. So you can feel more comfortable, because I did promise I'd talk about fish. So what we're going to do is use the virtual reality to control geometry by having virtual fish, so holograms floating in the tank with the real fish. And so here our virtual fish is just moving in a circle within the tank. The fish are less than a centimeter long. These are juvenile zebrafish. But this is just to show you that the real fish believes it to be in the volume with it, in the tank with it, even though we're only projecting onto the surface. Now, one problem with this technology, remember that when the paper was moved ever so slightly, the illusion was broken? That means I can't ever put two fish into this tank. Even if one is 1/4 of a centimeter away, it's like for it, the paper's moved, the world looks distorted. But what we can do, and what we have done, is we network the systems together, so this one can interact in real time with the hologram of the fish in this tank, and vice versa. And the great thing about virtual reality is we can control space and time. We can put in delays. We can, we were talking earlier about changing the water and even having different compounds in different tanks. But one of the things we published recently was showing that when you couple the systems in this way, the fish recover their schooling dynamics. And Iain went to great detail to show that the two fish in the matrix behave identically as if we take them out and look at how they behave in the real physical environment. So an important validation. We're building a whole bunch of these systems in Konstanz. So now let's think about the bifurcation story again, in the context of fish schooling. We take exactly the same model. All the systems we've studied, every one has got the same two parameters, right? Natural selection has found the sweet spot that allows them to make highly effective decisions. But now fish are not just attracted to static objects. They're really attracted to moving fish. So I'm just coloring them in red so you can see two virtual fish. And we predict that if we change the lateral distance smoothly, that changes the angle perceived, which should cause a bifurcation in the brain of the fish. And indeed, that's what we find. And similarly, if we look at three fish, now we see a double bifurcation. But it looks a bit weird because what's happening is we have a camera sort of following the fish. It's a moving frame of reference. And so if we didn't have the theory, we wouldn't know what experiment to do. And suddenly, even if we did this experiment and I got data that looked like that, I'd go, well, that looks weird. I would have no idea what to do with it. But because we had the theory, we could then interpret the data correctly. And we followed up with looking at all sorts of different staggered positions, tons and tons and tons of different configurations that I won't go into. And the model fits every single possible configuration. Now working with Arm and Bal, we're able to have a fictive preparation where we can go into the brain and have the animal, when it thinks to move left or thinks to move right or thinks to speed up, we decode that. And in the virtual world, that's what it does. So it can freely interact with the virtual fish, but we can record the brain at cellular level resolution. And that's the setup. And I won't go into details here, but if you're interested in this, we have this recent paper where we do tons more stuff, including experiments with 512 and 1024 mode deletion, which I saw today, showing that this is not just about zebrafish and it's not just about these tiny system sizes. And furthermore, this benefit of the algorithm, the ultra sensitivity, turns out to be very applicable to robotics, to making swarms of robots that make very effective decisions. So you can see here, we can scale up to different systems. And, if this is so robust, what about applying it in the wild? So with these baboons, we had GPS collars on the individuals, and we found tens of thousands of events where some go one way and some go the other way. So again, you can ask from a baboon's perspective, which direction does it choose as a function of the angle and the number of individuals going each way? And so we could test this bifurcation theory. If the top is where it's symmetrical, there's an equal number going each way, and this is due to the ultra sensitivity, even if there's a slight difference, you choose the majority. And that's indeed what we found within these wild unhabituated primates. And so coming back now to collective behavior. So this shows you that the brain is making decisions. The brain is a nonlinear dynamical system. It's not just the simple rules that we assumed back in the day. And so we thought, well, how can we actually use this potentially to make a difference, such as to explain locust plagues? Because they impact 1 in 10 people on the planet. And in fact, they're getting worse rather than better due to climate change. And you'll note here that these huge invasions, if you look at the date there, January 2020, right, what else was happening around then? You may remember, it was very hard to move people and equipment around Africa. In fact, our team got the second last plane out of Kenya before the airline stopped all flights due to COVID. And using our new technologies, we can track the animals in the field as well as do virtual reality experiments. And I won't go through these experiments, but suffice to say that it turns out that they're turning off olfaction, they're turning off tactile cues when in swarms. How incredibly convenient, because that means vision is the one thing we're very good at working with. And so we generated this virtual reality environment for locusts, where the locust is on a motion compensating ball. As the locust moves, the ball always moves to keep the locust on top, so it can march for 8 hours a day like it does in the wild. And by tracking the movement of the ball, we can then record the movement of the animal in a virtual swarm. And I can't show you what it's like to be a locust in a swarm because they've got pretty much 360-degree vision. And imagine these other locusts are floating in front of you, actually volumetric, like holograms in front of you. That's how it looks to the locust. And so we can design experiments that we would never have thought of without the cognitive theory, and we would never be able to do without the virtual reality. Such as if you use the classic model, like the Couzin model, it predicts that animals will just go with the flow. If you've got a swarm on one side and a swarm on the other side, you just go with the flow. The cognitive model, however, predicts, no, you should go orthogonal to the flow until you're within the group, and then you move with them, right? So we can put these models in contradiction with each other. So I showed you they walk on this locust motion compensating ball. And this is the best I can show, but imagine you can really see all the way around. So you can see these swarms moving in the same direction. And the cognitive theory predicts the brain decides to go this way or this way, rather than going with the flow. So contradiction to all previous models of collective behavior. And so this is the type of theory, and I'll just show you a subset of the data in the paper. We have tons of data. Now, that we were so excited about this. Now, we have a universal model that works for ants, flies, locusts, fish with exactly the same parameters. Great. So now I can come back to collective behavior. Because before I had these fixed targets, or I had moving targets. But what's really going to happen in the real world is I am inducing a bump in your ring attractor, but you're also inducing a bump on mine. And you, and you, and you. So now we have this recursive feedback. If each individual is, if other individuals are creating these bumps and then the brain is deciding consensus where to go, and you're doing the same thing, then maybe we have a new model for collective motion. And we've solved this long-lasting challenge that I've been studying for the last 30 years. And so I got really excited. Let's do that. Let's make each other targets. Each one is a target to each other, and we let the model run. Ah, bollocks. We got gravitational collapse. They just collapsed in on each other. So how can this model explain everything at the individual level, but then not explain collective motion? And so I was really, struggling with this for months. And then I was thinking to myself of work by my friend and colleague Nako Malinovsky on bats, which showed that bats actually have two ways of representing space. Remember when I talked to you at the very beginning about the compass in the brain? No model of collective behavior includes that compass. So, if I'm a schooling fish, yes, I will align or I'll move with other individuals, right? But it doesn't matter if I'm facing this way, this way, this way, right? But in the real world, I'll show you videos from the Maldives, the fish know which way the shore is. They know which way the deeper water is. They get information about the position of the sun. They have olfactory gradients in the environment. And we know that even flies have this ability. So we thought to ourselves, well, of course, this is really obvious. What if, like insects, fish are able to have this allocentric representation? So you have an environmental cue, irrespective of what it is. And it doesn't need to be the same between the different individuals. And we run the model again, and lo and behold, we suddenly get groups moving, turning, all the dynamics that we see in nature. And this came out two days ago, so this is like the very latest work. And what this shows is that you don't need these guessed rules, the rules I guessed back in the day, and the brains synchronize with each other to create collective motion. And so it's an entirely different understanding of how collective behavior is generated. And we were delighted when it was published in Science, and they acknowledged that it's rewritten the rules of the swarm. But I want to note that our fish work is lagging behind, but the fish experiments supported this long before the locusts, actually. We're just very slow at publishing it. And if you're interested, there's a great video by Tom Scott, the YouTuber, got almost 3 million views in 2 weeks. People are really interested in the stuff we're doing, and we want to get our science out before we publish. So we share our data, we share our experiments, and you can see some of these locust experiments. And we're developing for these non-model organisms like calcium imaging, and it's a great system. You can go from the neural dynamics up to continental scale behaviors. And in Konstanz, we have this amazing environment where we did experiments with up to 12,000 locusts that Tom Scott filmed. And this is showing you time lapse. And it's the first time ever anyone has got a swarm to move in a constant direction. And it's only if they have allocentric cues. And we can track the motion of all of the individuals as well. So all of this is happening just down the road in Konstanz. I'm sure many of you have been there. If not, it's a very beautiful historic city by the lake. We have these different disciplines working together. And we have a new building, it's about 2 years old, dedicated to the study of collective behavior across scales. And the imaging hangar is what I mentioned to you before. This is Tom Scott, just showing you a sense of scale. We can track a fruit fly at sub-millimeter precision, 500 hertz in this huge volume, not that we would, because fruit flies are terribly dull, but we can track animals in 3D and do all sorts of cool things. We can get 3D information from 2D, such as in these birds. But one of the really amazing things about the technologies we've been developing is that we can use them in the wild. And this is a study on the wild Indian grey wolf using the new version of T-Rex 2, which is about 2 weeks old. And I don't have a video of T-Rex 2. This is the old T-Rex. T-Rex 2 is much more sophisticated. You can identify animals with no markings. You can, therefore, you're not, if they move over each other, it doesn't matter. You still know who they are. You can do visual fields. You can do body posture. And importantly, it works in very complex environments, such as with the wolves. We're tracking them at day and night. T-Rex can identify the different wolves based on coat pattern differences. And also, we can get the trajectories of the animals in georeferenced coordinates. We have the 3D structure of the environment. But you can see from this body posture tracking, you can get behavioral information. This is zoomed in. We're tracking 15 wolves at the same time. But you can get very complex information about each individual. You can see we're tracking all of the animals. We can reconstruct visual fields, and these are critically endangered wolves. But many of you here are interested in aquatic environments, so we also work in aquatic environments using the same technologies. This is a 3D rig that we used in Israel and also in Australia to track octopus hunting. So everyone thinks octopus are solitary. This is the day octopus. And indeed, it's solitary unless they're mating, but they have very complex social lives, just not with other octopus, with different species of fish. They must always hunt together, and they have different roles within the group. But when we published this work, the only thing that the media was interested in is that we also discovered, as Stephen is going to express here and show some of our videos, that the octopus will also punch individuals that do not cooperate within these hunting groups. And he makes the very valid point that having 8 arms makes for a very effective boxer underwater. It was nice to make it onto Stephen's show before he gets cancelled next year. And you can see it's very stressful doing this work. This is our team in the Maldives. And this is studying shark-fish interactions that I showed you at the beginning. It's actually not just shark. There are also these jacks as well that hunt together with the sharks. And again, using T-Rex 2, we can track the individuals within the group, not just the predators. I'll show you in a moment. We can track the prey as well. And this is digitally zooming in. We're not actually zooming in, just to show you the details. And here we'll digitally zoom in again to show you can even see individual. Oh, I didn't mean to do that. I want to show you this because we can even show individual predation events. Both successful and unsuccessful. So what an amazing time to do science. And also it's a great way to do outreach to the public. And so August and Angela won a big photography competition from their work on this project. And so it got lots of attention into the science, like why are we doing this? Why are sharks important? Why is shark conservation important? And as I said, we can even track the individuals within the schools. So super exciting time to take our ideas into the wild. This is about 25 sharks hunting together. They also hunt collectively. Inside each of these vacuoles is a shark. And this is a time lapse. So again, a massively multi-scale problem, like going from the neural dynamics up to the individual decisions, up to the swarm of locusts. Here we can go from the microscopic interactions all the way up to hopefully ecosystem level questions. And it's also incredibly beautiful. And thanks a lot for listening. And because of this audience, in the background there, that's a real fish school in the Maldives in time lapse. Isn't that freaking cool? And we've got all of these data that would be very exciting to study. And thanks again for the invitation.
Thanks a lot, Iain, for this really nice presentation and all these super cool videos. Maybe some people already have some question around. Yeah.
Thank you very much. It was a very fantastic talk. Really enjoyed it. I was wondering in the bifurcation experiments, I didn't really understand what bias was introduced in the brain or at the individual level to choose one to the other if
Yeah.
identical.
Yeah, so that's a really important point, and in fact, that's why we do these experiments under identical conditions to see this decision-making process, because it's actually it's an old idea called Buridan's paradigm. Imagine an ass, Buridan's ass is hungry and thirsty, and there's water over there and there's food over there. If those two cancel each other out, then the poor animal is going to be sitting there in the middle. Or if an animal can't decide, you need to break indecision, right? And so what breaks that indecision is the fact that you have this negative feedback. So you're trying to extinguish the other opinion, and they're trying to extinguish you. You're trying to excite those around you to get them to extinguish the other opinion, and they're doing the same, right? And then just noise, just random noise. If the options are identical, random noise gets amplified due to remember I told you the ultra sensitivity? Just a little fluctuation of noise causes one just by chance to start winning, and then it will extinguish the other one. And if the two options are identical, it's 50-50. But it's very important you break that symmetry. Now, the interesting thing is, what if the options are a tiny little bit different? That same process that causes that decision-making allows them to exquisitely accurately detect a tiny difference that's not due to randomness, that's due to some systematic bias in the presence of randomness, and to make highly effective decisions. So the physics of the problem at that point really allows them to make fast and super accurate decisions. That's a great, great question.
And also, okay, so now I understand.
Yeah.
And so also the fact of introducing this allosteric component, this environmental cue,
Yeah.
that enables to fix.
Yeah, they need to have something. So if you do, I mean, fish in the dark don't do anything, at least the ones we've studied. But if you put a fly in the dark, it will keep walking, but then its heading direction gets disorganized. It doesn't really know which way it's walking, right? And so in our locust experiments, we did these experiments, and even though our lab, we tried to make it symmetrical, it turns out the lighting was slightly asymmetrical. And they used that alone to break the symmetry and to make these marching patterns. Now, if we put camo netting over that to break up any patterns, then for controlling for light level, they can't swarm, they can't march. And so really, when we study these fish in the Maldives, in the mornings, they come in from the reef into the shallows. And they're there all day. And then in the evenings, like the school will actually split every day, split into two. One goes that way, one goes that way. And we follow them as far as we can follow them before they go into the deep water. They know where they're going. We know fish can navigate. I mean, this idea that there are sort of simple automatons that, it's nonsense. And now we have very clear neurobiological evidence that they have a compass. Even in sterile lab conditions, they keep a compass going. I mean, it's amazing that the Drosophila do it without polarized light, without any of the things they normally have access to. But it's because it needs to be super robust. So they'll use many of these different modalities to lock their compass. Yeah. So, but astonishingly, this is the first model that's ever introduced that to collective behavior.
Thank you.
So there's no, and importantly, again, there's no alignment. The bumps just are the positions, they're the bearings towards other individuals. There's nothing you need more than that. It's in the navigational circuit already. For an animal to evolve to be collective, natural selection has to do very, very little.
Maybe I have a follow-up question on this. So if you were, for instance, studying differential conditioning in an animal, you will expect this critical point to just slowly disappear? Or it will just evolve to be a little...
No, this is a geometric principle. This will always exist as long as there's a geometric transition.
Okay.
Right, as they're going through this, so the angle here is, but I mean, of course, if the animal knows about the environment, if, for example, you have, let's for simplicity say 2 options, and it clearly knows this one is better, or it's been conditioned to know this one's better, of course it will just go straight to it.
Okay.
Right? This is only relevant in, well, I mean, decision-making theory in general is only interesting when decisions are difficult, right? Otherwise, you can just go straight to the options. So that's why we study it in difficult decision-making, but you'd expect natural selection, of course, to be, to want to make good decisions. And one of the things we're trying to study now is also sexual selection in this context. Where a decision really, really matters, who you choose to mate with.
Thanks for an awesome talk. Maybe a more speculative question. I wondered if you thought that there was something similar going on in the brain for time. So, if human, kind of, not to narrow the field, but, if I have 2 job offers, say, or, and I have a time limit on that, is there going to be a bifurcation moment that approach the time horizon?
So fascinating question, and there's two aspects that I'll talk about. We can chat about it later, maybe. So I was just at Columbia University in the neuroscience department there, and they're beginning. So I didn't mention, but for anyone that knows about decision theory, things like competing leaker accumulator models or drift diffusion models, we can map to this model mathematically formally. But this model explains thresholds as phase transitions rather than these abstract thresholds. But if you look at data from monkeys, now they're doing eye movements and so on, but the brain is still making decisions. The fast decisions, they go straight to the option in neural space, and we map ours to neural space too. And for slow decisions, they do the bifurcation, right? And so that's consistent with that. If either just by chance you want to make a fast decision or you're time constrained, you can force animals, to switch between these two different strategies. And we explain that in the paper that's in revision at the moment, how you can do that. But time gets even more interesting in another way, right? Movement through space, at least the spatial aspect of movement through space, is reversible. You can go back to where you've been before, but you can't reverse time. Time is unidirectional. So we've looked very little at time so far, but one of the great things about the virtual reality is, well, what we know is that I simplified it today, but the brain is actually not encoding space in a Euclidean way, right? The brain is actually warping space and probably warping space-time to make highly effective decisions. And that's what we're working on at the moment. We're working on a very general theory of how you warp space-time for optimal decision-making and how that can then map back mathematically to these models. So it's kind of crazy, but yeah, this is the big frontier, is how do animals represent space and time?
Maybe I have just one more, so you talked about this heading compass that you identify in zebrafish and other species.
Yeah.
I was wondering in zebrafish, what was the minimum age that you started to observe it?
So I want to emphasize that was not my work.
Okay.
That was the work of Ruben Portugues. I had the paper up there before. Now I guess he was working on 5-day-old fish because he was working in Germany, or 5 or 6-day-old fish. And I know that the Rowley lab in Tübingen also has since found also place cells and other. So it's I think fish basically have this from the very beginning. Now that does not mean it doesn't get more sophisticated and change. For example, one of the reasons I was so interested in fish is that they live in a 3-dimensional world, right? The ring attract I showed you is 2-dimensional. And the reason I showed you a 2-dimensional model, not because it doesn't work in 3D, it does, but the juvenile zebrafish, it doesn't matter how big your tank is, when they school, they're always schooling on the same plane. It could be either here, could be here, could be here. They prefer the surface mostly, but it can be anywhere. But they only ever school on the same plane. And to disassociate socially, it's in the science robotics paper actually, to disassociate socially, they move up or down a tiny bit. Half a centimeter up, half a centimeter down, they're completely unconnected to each other socially. So the way they represent X, Y is very different than the way they represent Z. And that I find fascinating. Is it a feature or a constraint? Because if you actually look at animal groups, the very few studies that have looked at 3D structure of fish schools find that most fish schools, if not all, are layered. There are stronger interconnections within the layer than between the layers. Theoretically, we've shown that that makes sense for information flow. So maybe it's a feature. But another possibility is as the zebrafish grows and its brain gets more sophisticated, it can then start representing 3D space in a different way. I don't know.
So you believe that one hypothesis could be that the schooling in zebrafish, for instance, is starting later in age because maybe their perception of the environment is not good enough?
I don't know. I mean, it starts very early. It actually starts earlier than people think, around 10, 11 days. But it's quasi 2D. They really don't. But I mean, are they not interacting when they move across a plane because they're choosing not to interact, or because they can't interact? There's a very different question. And I think it's because they choose not to interact. Because if they decide to, they can be over here and one over here. And if they decide to interact, this one can go all the way down here and then go into the plane and then they interact, right? So it's not that they're not seeing the other individual. They're seeing it, but they're not interacting with it, they're not schooling with it. Even if they're very close, they're not schooling with it. And so most people just do experiments in 2D. And so you would never see this. Or you'd never know whether your fish are connected socially or not connected, because even a small difference for a tiny little fish can be a huge difference in social connectivity.
Hi, I was wondering what in the brain structure or neural structure would determine whether a species would exhibit collective behavior or not?
So we think that, I sort of briefly mentioned it, we think that natural selection can just take the standard navigational circuit that all animals have, asocial animals as well, and by tuning it in a tiny little way, bang, you can get social behavior. It should be very, very easy for natural selection to turn an animal from social to asocial. Now, one very exciting and interesting example of that are animals that can switch between asocial and social. And the best example is the locust. So I didn't have time to explain it, but locusts can't stand being near each other. They're shy, cryptic green grasshoppers. It's only when they're forced to come together within an hour, they now are social, right? If you separate them again, they become asocial. And so the brain switches between these different states. And so we don't think this needs a major reorganization of the brain at all, which is why it can be so fast. It can be neuroimmunomodulation to a large degree. But this is one of the things we're studying. And so, yeah, it really has implications for the evolution of group living and the evolution of collective behavior. Because tiny tweaks, remember I showed you what happens when, how the brain responds when there's no sensory input and you get things like zigzags and spirals. That's just changing these parameters that natural selection can change with neuromodulation, like how excitatory is the system, for example. And so we think that the brain is this complex dynamical system that natural selection, well, firstly, we also show in that paper that if an option appears, the brain will suddenly exhibit a phase transition into the decision-making regime. When the option disappears, they can do a phase transition back into search. You don't need to add that in. The animal doesn't need to make it is a decision, but it's a spontaneous emergent property of the neural networks. So I think I hope it has lots of implications going down the line, but this is a hypothesis at the moment, based on proper neural data, but we still need to do much, much more work to really test this hypothesis.
I think we have a question on Zoom, so you can ask it.
Yes, thank you. Thank you for a very nice talk. I was just out of curiosity wondering, because you talk of different systems that all of them use some form of visual cues, where these collective behaviors in movement are unique of diurnal organisms, or do we have any animals that are nocturnal and also show these things, and then what other then systems would be more important there?
Great question. I would turn to fish for this question because half of all vertebrates are fish, and you can pretty much find a fish that can solve all sorts of different problems. And so the fish we have studied have tended to be small freshwater fish, tropical or local, because they're easy for us to collect and so on. And they are diurnal, and at night they can't school. There are some species of fish that seem to be able to school in the dark. And so they could use, for example, hydrodynamic cues. There's some brilliant studies on hunting catfish that can actually, when they're hunting prey, they detect the trail of wakes, even 20, 30 seconds, maybe even a minute later, they can detect this wake and follow it, right? I'm not saying they're using the lateral line, but they are detecting these socially generated flows. When I put the larger fish in the dark, remember they still schooled together. So I think for small fish, like zebrafish and small fish, they're kind of screwed because there's no way you can use hydrodynamics at that scale because of the viscosity of the water. So they really have to use vision pretty much exclusively. But when you get to larger fish, or maybe even large schools of fish that can move water collectively, then there's possibilities that they may be able to school without vision. But we and others haven't really studied that yet. Blind cavefish don't school, unfortunately. So there are things like lanternfish. They produce flashes of light and they school that way. Daniela translucida produces acoustic cues, not for schooling, but for mating and so on. So you could use acoustics as well. So the crickets we're studying, for example, they interact acoustically. And many fish produce sounds. Many fish can produce sound. So that's a possibility too. So, I often think, it takes us years and years to work on one species of fish, and it just tells us about that one species of fish. We hope it's general, but realistically, the diversity out there is so great that it'd be so fun for people to work on non-model systems that behave very differently.